Welcome to The Explainer. Today we're doing a real deep dive, a masterclass if you will, on a super important neurotransmitter and hormone, 5-hydroxytryptamine, but you probably know it by its more famous name, serotonin.
All right, so let's jump right into this. We're going to follow the entire life story of a single serotonin molecule. I mean, from what it's made of, to how our bodies create it and put it to work, all the way to how medicine can, step in and interact with it to change our health. It's a fascinating journey.
So here's the game plan. We'll start with the basics. What is serotonin? Then we'll cover its life cycle, its many jobs in the body, and jump into pharmacology. After that, we'll see what happens when the system goes haywire, and we'll wrap up by looking at some related peptides and pathways. Let's get started. All right, first up, let's get the fundamentals down. What is this stuff? And, where do we actually find it in the body? Okay, so this is where things get really interesting right off the bat. You probably think of serotonin and immediately think brain or mood, right? Well, get this. A whopping 90% of it is actually in your gut, in what are called enterochromaffin cells. Only the last 10% is split between your platelets and, your brain. And it's not just hanging out in these places. It's got specific jobs to do. In the gut, it's all about digestion and gut function. In your blood platelets, well, they have this really cool high-affinity uptake mechanism. They basically snatch up serotonin as it floats by in the gut circulation, and that helps them clump together. And then, of course, in the brain, it's a major inhibitory neurotransmitter. Think of it like a brake pedal for things like mood and pain. And here's a fun fact. It was first discovered because of its effect on blood. Scientists noticed it was a substance released from blood clots that constricted blood vessels, a vasoconstrictor. That was all the way back in 1951. All right, so we know where it is, but how does our body actually make this stuff? And how does it get rid of it? Let's trace the entire life cycle of a serotonin molecule, from the moment it's created to the moment it's broken down.
Okay, this chemical pathway might look a little intimidating, but I promise it's not so bad. It's a whole journey, and believe it or not, it all starts with something you eat, specifically an amino acid called L-tryptophan.
So the main thing to remember is this. It's a two-step process to build it and a two-step process to break it down. Super symmetrical. Think of it like an assembly line. Step 1. An enzyme called tryptophan hydroxylase gets the ball rolling, turning tryptophan into something called 5-HTP. Step 2, another enzyme, tryptophan decarboxylase, does the final conversion, and boom, you've got serotonin, or 5-HT. Now for the cleanup crew. Step 3, an enzyme you've probably heard of, monoamine oxidase, or MAO, starts the deactivation process. And step 4, aldehyde dehydroxylase comes in to finish the job, turning it into the final waste product, 5-HIAA.
And that final waste product, 5-HIAA, is actually super useful for doctors. By measuring how much of it is in your urine, they can get a really good idea of how much serotonin your body is making. And because we understand this whole process so well, we can actually step in and control it. There's a drug called telotrestat, for example, that literally blocks that very first enzyme in the assembly line, tryptophan hydroxylase. It just shuts down production at the source.
Okay, so that's the chemistry. But let's see this in action where it really counts. In the synapse. That's the tiny gap between two nerve cells. It's basically a 6-step signaling process. First, serotonin is made inside the sending neuron. Second, it gets packed up into little bubbles called vesicles for storage. Third, a nerve impulse comes along and says go, releasing it into the synapse. Fourth, it floats across and binds to receptors on the receiving neuron, delivering the message. Fifth, and this is super important, it gets cleared out of the synapse. And sixth, any leftovers are broken down into that 5-HIAA we talked about.
Now that fifth step, clearing the synapse, is absolutely critical. You can't just have the signal firing forever, and the body has 3 ways to do this. The main way, the big one, is reuptake. There's a special transporter protein called SERT that acts like a tiny vacuum cleaner, sucking the serotonin back up into the sending neuron to be used again. And that is exactly what SSRI antidepressants target. They block that vacuum, leaving more serotonin hanging around in the synapse to do its job. The other 2, less common ways, are for nearby support cells to just absorb some of it, or for it to simply drift away.
So, you might be wondering, how can one molecule do so many different things? The answer is receptors. Serotonin is like a master key that can open a whole bunch of different locks, and each lock triggers a different response. So let's take a look at these different receptors. And whoa, there are a lot of them. We're talking 7 main classes, which break down into at least 13 further subtypes. But here's the absolute most important thing to remember. Almost all of them, classes 1, 2, 4, 5, 6, and 7, are something called G-protein coupled. Think of these as dimmer switches. They're a bit slower and trigger a more complex chain reaction inside the cell. But then you have the odd one out, 5-HT3. This one is different. It's a ligand-gated ion channel. This is like a simple on-off switch. When serotonin hits it, a gate flies open, ions rush in, and you get a super-fast direct signal. That speed difference is everything.
So, in the nervous system, serotonin's influence is just huge. The neurons that make it are located in a central spot called the raphe nuclei. And from there, they branch out to pretty much the entire central nervous system, acting as that general inhibitory brake. But the specific effect all depends on which receptor gets activated. Remember that super fast 5-HT3 receptor, the on-off switch? Well, that's the one involved in pain perception. It's also a major player in the vomiting reflex, both in the gut and in the brain's vomiting center. This is a huge deal for chemotherapy patients, since chemo can trigger a big release of serotonin in the gut. Meanwhile, other receptors, like 5-HT1B and 5-HT4, are working in the gut's own nervous system. It's like a second brain, controlling its movements. And in a totally different role, the pineal gland actually uses serotonin as the raw material to make melatonin.
Yeah, let's stick with that melatonin idea for a second. It's the hormone our body releases at night to manage our circadian rhythms, our sleep-wake cycle. And just like serotonin, it has its own set of receptors. MT1 and MT2 are the main ones. And of course, once we know that, we can make drugs to target them. So you've got drugs like remeltation and tesamortation, which are agonists, meaning they activate those receptors to help with sleep. Then there's another one, agomelatine, which is a bit more complex. It's an agonist for the melatonin receptors, but it also weakly blocks a couple of serotonin receptors. And that combination is used to treat depression.
Okay, this is pretty wild. If you were to inject serotonin directly into someone's veins, you'd see this really distinct three-part effect on their blood pressure. And what's so cool is that each phase is perfectly explained by a different receptor subtype. First, you get a sharp, immediate drop in heart rate and blood pressure. That's the fast-acting 5-HT3 receptors doing their thing. Then, just for a moment, you get a rise in blood pressure. That's the 5-HT2A receptors causing vasoconstriction. And finally, you get a long, sustained fall in blood pressure. That's because 5-HT2C receptors are causing the arterioles in your skeletal muscles to dilate.
So what about the rest of the body? Well, in the cardiovascular system, it's a powerful vasoconstrictor, except for the vessels in your skeletal muscle and heart. There, it actually dilates them. Weird, right? In your gut, it's a major stimulant for smooth muscle, really increasing the tone and peristalsis. That's the squeezing motion that moves food along. This is actually why certain tumors that pump out way too much serotonin cause severe diarrhea. In your lungs, it has a small stimulant effect on the muscles there. And with platelets, it causes them to aggregate or clump together. But here's another one of those fascinating twists. If the lining of your blood vessel is healthy and intact, it actually leads to vasodilation. But if that lining is damaged, it causes constriction, which can make things like vascular disease even worse.
Okay, so now that we've got all that biology down, let's get to the really practical part, pharmacology. Because we understand this whole system, the life cycle, the receptors, we can design incredibly smart drugs that can tweak the system to treat all sorts of conditions. Let's start with the agonists. These are drugs that pretend to be serotonin, activating the receptors. And you can see how specific they are. For migraines, you have the famous triptans like sumatriptan, which hit the 5-HT1D and 1B receptors, or a newer one, lasmicidone, which is very selective for the 5-HT1F receptor. For anxiety, you've got buspirone, a 5-HT1A agonist. For weight management, there's lorcaserin, targeting 5-HT2C. And then you have drugs that do two things at once, like flumanzerin, which activates 5-HT1A while blocking 5-HT2A.
All right, so if agonists turn things on, antagonists turn them off. They block the receptors. These are your go-to drugs when you have too much serotonin activity. The classic example here is ondansetron. It blocks those fast 5-HT3 receptors, which, as we learned, are key for the nausea reflex. That's why it's a game changer for people undergoing chemotherapy. Then you have other blockers like ciproheptadine, ketanserin, and ritidanserin, which all focus on blocking the 5-HT2 receptors. So serotonin is clearly essential, but you can definitely have too much of a good thing. And when it comes to serotonin, too much can be downright dangerous. So let's look at what happens when the system goes wrong, starting with serotonin excess and then a really interesting historical poisoning.
This brings us to a very serious, potentially fatal condition called serotonin syndrome. It's basically an overdose of serotonin activity, and it can happen for a bunch of reasons. Taking MAO inhibitors, SSRIs, amphetamines, you name it, especially in combination. The symptoms are the key thing to know, and there's a great mnemonic for it: shivers. It stands for shivering, hyperreflexia, that's overactive reflexes, increased temperature, vital sign instability, encephalopathy, which is just a fancy word for altered consciousness, restlessness, and sweating. Shivers. Really important to remember.
And this connects to a really bizarre historical condition called ergotism, or St. Anthony's fire. It's a type of poisoning you get from eating grain, like rye, that's been infected with a fungus called ergot. The chemicals in this fungus, these alkaloids, mess with all sorts of receptors: serotonin, dopamine, adrenoceptors. Some of them, like ergotamine, cause severe vasoconstriction, while others, like methosergide, can cause tissue fibrosis. It's nasty stuff. But in a classic pharmacology story, we've managed to modify some of these poisons into useful medicine.
Okay, for our last section, let's zoom out a bit, because to really get serotonin, you have to understand that it's not acting alone. It's part of a huge, complex network of signaling molecules. So let's just briefly touch on some other peptides and pathways that it works with.
Right, so take migraines. For years, we focused on serotonin with the triptans, but now a whole new class of drugs targets a different peptide entirely, called CGRP. It's a huge deal. Same for nausea. We have the 5-HT3 blockers, but we also have drugs like aprepitant that block a totally different thing called substance P, and the list goes on. Neuropeptide Y, vasoactive intestinal polypeptide, it's all part of this massive, interconnected chemical conversation happening in our bodies.
And just to drive home how these cascades work, let's look at another classic one, the renin-angiotensin system. This is the master regulator of blood pressure, and just like serotonin production, it's a step-by-step process run by enzymes. First, an enzyme called renin kicks things off, turning angiotensinogen into angiotensin 1. Then, another famous enzyme, ACE, converts that into the really powerful angiotensin 2. Angi2 then acts on receptors to constrict or dilate vessels. But there's another step. A different enzyme, ACE2, can convert angiotensin 2 into something called Ang017, which has a totally different effect.
And this slide really shows a beautiful principle of biology: balance. It's like a yin and a yang. On one side, you have angiotensin 2, which is generally bad news for tissues. It causes constriction, inflammation, fibrosis. On the other side, you have its breakdown product, angio1-7, which is protective. It's anti-inflammatory, anti-fibrotic. This idea of opposing forces within a single pathway is just fundamental to how our bodies work.
So, there you have it, the complete story of serotonin, from a single molecule all the way to its role in complex medicine. But it leaves us with a really exciting question, doesn't it? We've identified at least 14 different receptor subtypes, and we only have drugs for a handful of them. So you have to wonder, given its incredible reach, from our gut to our brain, what amazing future therapies are still locked away, just waiting for us to find the right key for the right receptor?